Okay. So we're starting our new unit today. I did literally like 3 minutes ago enter your exam grades. I have them with me, so if you want to check them out at the end of class, you can. I didn't bring the key with me though, because I literally just wanted to get the grades in. So we can spend time going over them in lab next week, because we already had lab this week. We'll spend time in lab next week going over them, looking at the right answers, kind of going through it like that, if you like. Today we are starting Unit 3, which is all about the nervous system. In fact, we will be in the nervous system for the rest of the semester. That's where we're chilling, all up until the middle of December. That's what we'll be looking at, the brain. We're going to start with kind of the basics, a nice overview of how the nervous system's put together, and neurons and all of that stuff. So we'll kind of go into neural tissue and such. There we go. All right, so overall, when we're talking about the nervous system, we're talking about our voluntary movements and all of our sensations, right? So we have sensations coming in, movements, the commands for the movements going out. So our nervous system directs all of that, right? And our nervous system also receives all of that input. So here we're talking about regulating respiratory rate based on incoming information, right? Based on all of the sensory information coming in. Regulating blood pressure, again, based on the sensory information coming in. Regulating body temperature, pH, right? Hormone levels, sleep-wake cycles. All of these things are all regulated from our nervous system. So we're going to look at how that happens, so the physiology piece. And we're going to look mostly at the anatomy in lab, right? After we get done muscles in lab, we'll spend time looking at the spinal cord model and the brain model and the neuron model and look at those and how they're put together. But we'll name all the parts in here at least, right? So it all kind of fits together. So our nervous system is kind of divided into two functional pieces here. So anatomically, we can divide our nervous system into the central nervous system and the peripheral. So the peripheral nervous system, this is everything that's in our arms and legs. This is basically everything outside of the central nervous system. The central nervous system is our brain and spinal cord. Our peripheral nervous system is all the nerves. So our peripheral nervous system is right now bringing information into the brain about, how your pencil feels in your hand, the temperature of this room, right? Whether or not your seat was warm when you sat on it. All those things going into your brain, right? Going from peripheral to central. So information flows in two directions. Information can flow from the peripheral to the central, right? This is all sensory information. Right? All of the sensation stuff is going in from the peripheral nervous system into the central nervous system. And then what leaves the central nervous system and goes to the peripheral nervous system? What is it? Something. Example. You touch a hot stove. You feel it, that sensory information's coming in. What do you do in response to touching that hot stove? You pull your hand away. So what information's coming from the central nervous system? It's a motor command. Right? A motor command is telling your flexors to flex, to pull away that hand. So motor commands come out. Right? And that's this big overarching theme in the physiology. This is how your brain controls things. Millions of neurons involved in your brain and spinal cord. Of course, your brain's protected by the skull. Your spinal cord's protected by the vertebrae, right? And nerve cells and neurons are interchangeable. You can use those interchangeably. They're the same thing. So you can call it a nerve cell, or you could call it a neuron, potato, doesn't matter. So the nerves themselves are made of this complex structure. We've kind of looked at them briefly before, All the nerves outside of our brain and spinal cord are in that peripheral nervous system, right? So they have those long axons. Most of the cell bodies actually come from the central nervous system and then go out into the periphery. In other words, the cell bodies are kind of rooted in your brain or rooted in your spinal cord, and then the axons, these long extensions, come out and they form these nerves. The nerves that go down and contact all of our skeletal muscles. So we actually have 12 pairs of nerves that come directly off of the brain. They're called cranial nerves, right? So I like to draw this. My gingerbread man of nerves. I know it's horrible, but bear with me. It does the job. Let me grab some colors here. So we'll put our brain up where it belongs, right, in our spinal cord. Brain and spinal cord, that's our central nervous system. Our brain has 12 pairs of nerves coming off of it. And so by pairs, literally, they're going to the right side and to the left side. These are 12 pairs of cranial nerves. So we call them cranial nerves because they're coming right off of the brain. Our spinal cord has 31 pairs of nerves coming off of it. We call them spinal nerves. Right? And so these nerves, I drew them short, but really, I mean, they're going, right? They're going everywhere. Right? They kind of go up, they control. So these are neurons that are bringing in sensory information and carrying out motor commands, right? This kind of ebb and flow of information to and from the central nervous system. So that's the picture that I just drew, right? But this one looks a lot better than my little gingerbread man over there. But you get the idea, right? Peripheral nervous system is all of these nerves. So all of the blue and the red is peripheral. The black is the central nervous system. All right, so let's talk about these sensory nerves. So sensory nerves are the nerves that carry information from the peripheral nervous system to the central nervous system, right? They're bringing information in all day long. Your brain is getting so bombarded with sensory information, it starts ignoring some of it, right? I bet you're not thinking about the way your shoes feel on your feet right now. You are now that I said it, but you weren't before I mentioned it, unless they were really uncomfortable, right? You're probably not thinking about the way your elbow feels on the table, right? It's all of this sensory information coming in, and your brain has to figure out, is that important or not important? What needs my immediate attention? What do I need to respond to immediately? Elbow on the table? I don't care, right? Shoes on your feet? I don't care, right? What Jamie's saying? Oh yeah, I better listen, right? That's the important stuff, right? Your brain has to figure that out. So our sensory division of our nervous system can be broken into two categories, somatic and visceral. So this is the functional division. Okay, so I'm going to erase this. This is the anatomical divisions, right? We can break them down based on where they are anatomically. They're either outside of the central nervous system or in the central nervous system, right? It's either the brain and spinal cord or it's not. That's the anatomical divisions. But our functional division is different. So our functional divisions we have sensory. And then of our sensory division, we have somatic sensory, and we have visceral. So when you hear the word somatic, you should automatically think of something that is conscious, something that you're aware of, right? It might even be something that you can consciously, voluntarily control. Somatic means you're aware of it and you know it's happening. You can control your reaction to it, right? Visceral's the opposite. When you think visceral, think like inside. This is like your guts, right? You don't have control over those things, but you have visceral sensory receptors in your stomach right now saying, we haven't eaten in a while. Maybe we should grumble so we eat after class, right? Or, whoa, you ate a big salad, right? You need to move this along so we can digest the fiber, right? Whatever the signals are, right? Your stomach is figuring it out, right? And so that's all visceral sensory. Somatic sensory, this is all stuff that you're aware of. So this is information about your body position. You know that you're sitting, you know that you're standing, you know if you're lying down, you know where your body parts are, you know if your legs are crossed, arms are crossed, whatever, right? This is all of our skin, right? All of our senses, touch, right? Temperature, all of those things. This is also what we call our special senses. So special senses are ones that we have a special organ for, like vision, hearing, balance, smell, taste, right? So those are all be considered somatic senses. Visceral, this is inside stuff. This is sensory information about the partial pressure of carbon dioxide in your blood, right? This is information about your blood pH. This is information about how much glucose you have in your blood, right? All of those things, all of that internal stuff. You're not consciously aware of it. I'll say subconscious, not unconscious. How about that? This is your subconscious. So, our central nervous system, the job of our central nervous system is to take all of this information in and integrate it. We call it an integrative center. It's figuring out what's important, sorting through the baloney to pull out the good stuff. You guys have to be good at this, right? As students, you have to figure out exactly what the instructor thinks is important, what they're going to test you on, so you can ignore all of the other junk and focus on the important stuff. It just so happens everything in this class is the important stuff, but in most classes, that's how it works, right? You're going to sort through that material and get to the important stuff. That's exactly what your brain does. Your brain has to figure out what is important, and you know what? 99% of the information coming in is completely ignored by your brain. Your brain doesn't respond to it, right? It'd be like sensory overload, right? Have you ever been in those situations where it's really loud, there's lots of lights, there's lots of people, there's lots of sensations, and you just want to get out of there? Oh, this is too much for me. Like Christmas shopping, that's how it is for me. Like going to a crowded mall, I'm like, get me out of here. Nope. Amazon Prime that. So, our motor division is our other functional division. Our motor division, this is carrying our motor commands, right? So here we have, again, somatic and autonomic. So we had somatic and visceral sensory, and just have a different word, but it's the same concept. Somatic motor, these are our skeletal muscles, right? These are all the ones that we're consciously controlling, that we're consciously aware of. The autonomic kind of relates to the visceral. These are the things that we're not consciously aware of. They're doing their thing. We don't control them. This is like the heart and smooth muscle. Bless you. The autonomic. This is all the smooth muscle and the cardiac muscle. So the motor neurons carry the motor commands to the skeletal muscle or to the smooth muscle or to the cardiac muscle, telling them to contract or do whatever, right? Contract or relax. The muscle itself is called an effector. We've used the word effector before when we talked about homeostasis, right? You have a stimulus that's detected by a receptor, that information goes to a control center, the control center directs an effector to change and turn off the stimulus, right? That's the homeostasis loop. The effector is the thing that's responding. So in this case, the effectors are the actual muscles themselves, right? The skeletal muscle. That's the effector. It's the thing that's carrying out the command. So we call these divisions by two different names. We use the word afferent or afferent. I usually say afferent. That way I pronounce the A and I recognize that it's an A. Or efferent. So when you see the word afferent, you should think sensory. Efferent, you should think of motor. You could also say the word afferent instead of efferent. And when you say think of like a relaxing back rub or a massage or a bubble bath or whatever. And think about your senses, like being happy. Afferent, right? Afferent, sensory. Maybe make that connection between the words there. Efferent, maybe think of the word like efficient. If you're efficient, you're probably moving around a lot and getting a lot of stuff done. Efferent, efficient, motor. Afferent, sensory. Afferent. I'm just trying to help you remember the words. All right. So on our motor division, our somatic motor division, this is looking at all of our voluntary stuff. Autonomic is all of our involuntary stuff. So this is all of that visceral, lungs, heart, etc. All right, that's just showing you information coming in. Is it spazzing out, or what does that mean? Information coming in and information going out. Okay. So, we did that. Yep, I think it's me. It's definitely not me. Okay. So this is the graphic organizer that we just created, right? All of this integrates the central nervous system and the peripheral nervous system into sensory and motor divisions. So let's talk about how neurons are put together. So that's this is kind of big picture stuff, right? Kind of just like how we did in muscles. We had this big overarching, we know that we're going to contract, we just don't know how we're going to do it yet. That's kind of where we're building to. We know that all this stuff happens. We know that this control happens. We know there's communication between central and peripheral nervous systems. Sensory information comes in, motor information comes out. We just don't know how it happens. So let's break it down and start looking at the structure of neurons and how they're put together. So all of our neurons have the same basic structure, although they're not all the same. There are some variations. They have something called a soma, or a cell body. So this is just going to be the structure of a neuron. So the cell body, this is where the nucleus would be, right? This is also where these little tiny clusters of endoplasmic reticulum are, and they're called Nissel bodies. On the model in lab, these are little purple things, and they're called Nissel bodies. They're just clusters of ER. Right. There's lots of mitochondria in there to help provide the energy for doing all of the things that our neuron has to do. There's a really complex cytoskeleton. Cytoskeleton's that like inner cobweb that serves as a network for things to be transported throughout the neuron. There's something called neurofibrils. Remember myofibrils? So here's neurofibrils, right? Neurofibrils do the same thing. This kind of acts as almost like a conveyor belt going down the axon. So coming off of the cell body is a really large process called the axon. We've seen it already in lab, looking at that lab model. But before we talk about the axon, let's talk about what else comes off of the cell body. There are little branches called dendrites. And these dendrites connect this cell to other cells. At the end of each dendrite can be a whole nother neuron. So the neuron that I just drew here could be connected to 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18 other neurons. In other words, it can be receiving information from all of those. Sorry, I forgot to turn my ringer down. Hopefully that works. Okay. The axon hillock is this funnel area, right? It's kind of bridging the gap between the cell body and the axon. It's this funnel area. I'll kind of draw like a green box around it. Axon axon collaterals are other branches that might come off of the axon. Right, that would be an example of a collateral. And the telodendria are the small branches that kind of jump from collateral to collateral. So there might be other little branches down here called telodendria. Ultimately, we're getting to the thing that we're actually familiar with, which is that synaptic bulb, right? At the very end, the axon terminal, right? We've seen that before at the neuromuscular junction. It's the same thing, axon terminal, synaptic bulb. It's the same word. It's like neuron or nerve cell. Doesn't matter what you call it, right? The purpose of this axon terminal is to communicate with the cell that comes next. Boop. I mean, love this board. All right. The Akzo Lemma is the extraplasma membrane that surrounds the axon. It's highly excitable. So it's kind of like the sarcolemma, but it's called the Akzo Lemma because it's the specialized cell membrane around the axon. It's designed to conduct electrical impulses. All right. So the Akzo Lemma is the cell membrane down the axon. Because what's going to happen, we've learned a little bit about action potentials already. The action potentials are going to come in from other cells. Our cell's going to decide, is this important? Do I need to respond? And then it's going to send an action potential down the axon, and then neurotransmitter is released, communicates to the next cell, over, so on and so forth. That's where we're going with this. The transport down the axon can be fast or slow. When it is slow, it's going to transport things from the cell body down the axon pretty slowly. However, it can happen quickly. And by axon transport, I'm literally talking about, like, imagine one of those, like, people movers at, like, the airport. You know, you just stand on it and it, like, moves you. Those things. That's what this axon transport is. So imagine that there's little guys down here that need, that need something, right? The way they're going to get it is by something shipping, right, like a little boat or a little mover, right, going down that axon. And so they can happen quickly or slowly, and that transport usually does not go the other way. It only goes from the cell body down to the axon terminal. It never goes up. Usually uses a lot of ATP to do that because it's a very long way, so there's lots of energy that's going to be put into moving things down. There's actual pictures of real neurons, so you can see how extensive they are, right? Kind of jumping in all directions, connecting. And this obviously is the much easier one to look at, the cartoony one, right? So all of these yellow branches are the dendrites. Cell body, Nissel bodies are those purple things in there. Axon Hillock, then our axon. I'm not even touching it. Anyways, it goes down to our axon terminals, which the axon terminals are touching our postsynaptic cell, right? So this neuron is communicating to maybe some gland cell. Say, hey, you know, you need to secrete some sweat right now, or secrete some oil or hormone or whatever, and so that's what those cells are doing. So let's talk about some disorders related to this. Polio Mellitus, right? This is the polio virus, right? This is impacting nerves, especially of the spinal cord. There's no cure, right? But it can be prevented through vaccination, and it's caused by a virus. So this virus actually goes into muscle cells, right? And it jumps from muscle cells to the nerve cell. And it goes up the nerve cell, right? So it literally goes backwards. It goes in this retrograde fashion. It goes from the muscle cell up. And so until eventually it reaches the spinal cord, right? And that's where it can cause paralysis. There's other viruses that use this same method, kind of infecting one cell and then jumping to the nervous system from those cells. Kind of a sneaky strategy. Herpes is one of those. Rabies is another one. Viruses that initially infect one part of the body but then find their way into the nervous system by jumping onto the axon and going up. They're like climbing up the sliding board. right? It's one of those days that I love this one. Oh. All right. So our neurons have three major regions. A receptive region, that's the region that's going to respond to an impulse, or respond to a command from another cell. Right? The conducting region, which is going to pass that signal on, and then the secretory region, the region that's secreting. Right? So those regions make sense based on their function. But not all neurons are the same. So what we just drew here would be considered a multipolar neuron, minus this collateral there. This would be considered a multipolar neuron. This is what most of your motor neurons look like, but not all of your neurons look like this. Some of them are bipolar, some of them are unipolar or pseudo-unipolar. So bipolar would be where we have a cell, an axon, a cell body, and a dendrite, like all in one line. Right? A unipolar would be where you have these cells and it's the same type of setup, but the cell body's off to the side. So instead of having all of these branches, it has one dendrite, one axon, and the cell body's either to the side or in the middle. This would be bipolar. This is found in certain special sense organs, like your eyes. Your eyes have bipolar cells. And unipolar cells are also found in special sense areas. Bless you. Actually, a lot of your sensory neurons are unipolar as well. Yeah, sensory neurons are. So, to kind of give you an idea of these unipolar. So the nerves that are throughout your peripheral nervous system, right? The cell bodies usually cluster around each other, right? So you can imagine a bunch of these all right next to each other, right? Because these are all the sensory nerves of one particular area of your body. Well, imagine that these are the sensory neurons of your arm. And I bet you've hit this spot before, and it tingles and it feels weird. Where am I talking about? On your arm. Yeah, underneath that medial epicondyle, hey bones, right? Underneath that is this really sensitive area that if you zing it, like stick your thumb up there, hit it on accident or on purpose, it tingles. What you're hitting when you hit that and you feel that kind of weird numb sensation, you're hitting a bunch of sensory nerve cell bodies. That's where they are. They're unipolar neurons. Half of them are extending this way, the other half are going up your arm, communicating with your central nervous system. They're all clustered right there. So most of our sensory nerves are the unipolar shape. So we also have functional groups. So those were structural groups, right? Multipolar, unipolar, bipolar, structural groups. But we can group them by function also. So we've already talked about sensory neurons, right? Sensory neurons are carrying information from the periphery to the central nervous system. We've talked about motor neurons carrying information from the central nervous system to the periphery. Interneurons are in between. Interneurons are within the central nervous system. These are the decision makers. These are the ones that decide, we're going to flex the forearm. We're going to, flex the hamstring. We're going to move our head. We're going to do this movement or that movement. The interneurons are the ones that decide. They're like little mini brains. Right? They take all the sensory information that's coming to them and they make the decision. Do we need to respond or not? And how to respond. All right, so they're relaying information between the central nervous system and periphery. Most of them are multipolar, right? So most of them are looking like this. And this is the way most of your neurons look. Just a few of them will look like that. So the flow of information looks like this. Right, sensory, well, we can start over here. Receptor, we haven't talked about receptors yet, but we always have a receptor. The receptor is going to communicate with a sensory neuron. The sensory neuron is going to communicate with an interneuron. The interneuron will communicate with a motor neuron. And the motor neuron communicates with an effector. Right, that's the flow of information. So this interneuron is in the central nervous system. So this is in the brain or spinal cord. Everything else is happening in the periphery. The decision making, the response initiation starts in the central nervous system. So that's just a picture of what they look like. All right. So there's a couple other terms that we should understand. One is in the central nervous system, we call this cluster of cell bodies. We don't call it a cluster of cell bodies. We call it a nuclei, which is really hard to remember because we're so used to thinking of a nuclei as this, right? We're so used to thinking of a nuclei as the control center of a cell. But in nervous system world, a nuclei is a cluster of cell bodies in your central nervous system. Bless you. Why do you think cell bodies would like to be clustered around each other instead of randomly spaced out? What do you think? Go ahead.
So they can like send messages to each other?
Yeah, so they can maybe communicate to each other, hey, this is what I'm doing. Is that what you're doing? Hey, this is what I'm doing. Yeah, totally. Why else? Have you ever seen like an electrical panel or like a motherboard of like some circuit, right? All of the circuits come from the same general area. They're right next to each other, right? All of the electricity in your house comes in through one wire and goes to an electrical panel. It's probably in your basement, right, or in a closet somewhere. That electrical panel is where all of the wires of your entire house come from, right? They don't come from here and there and there and there and there, because that would be really disorganized. It'd be coming from everywhere. How would you keep track of it? So if the cell bodies are clustered together, it keeps them organized. It's a way for your body to kind of regulate and control and keep up with quick changes, right? So we call those clusters of cell bodies nuclei when they're in the brain and spinal cord. When they're in the periphery, like this right here, we don't call it a nuclei. We call it a ganglia, right? Think of it like a gang. It's like a gang of cell bodies hanging out there. Hey man, what are we gonna do today? Right? Ganglia. In our central nervous system, we call nerve fibers, bundles of nerve fibers, we call them tracks. So we have tracks. Tracks going up, tracks going down, right? Tracks coming into the brain, tracks going out of the brain. Tracks going up the spinal cord, tracks going down the spinal cord. We call them tracks. They're bundles of axons, bundles of nerve fibers. In the peripheral nervous system, we call them nerves, right? We had 12 pairs of cranial nerves, we had 31 pairs of spinal nerves. We call them nerves, not tracks. But they're really bundles of axons, right? So we're saying that it's a nerve, but really it's a bunch of them. It's a bunch of these. A bunch of these together make up a nerve. So, neuroglial cells are these supporting cells. You can't have all of these fancy cells that do all this fancy conduction work, regulation, without them being supported in some way, right? You can't be a movie star without an entourage, right? You need someone there to do your hair, do your nails, make sure your outfit's on point, right? And that's what these neuroglial cells do. They're there to nourish, protect, right? And make sure that these nerve cells have exactly what they need, right? They'll help replace the neuron if it is able to be replaced. But neuroglial cells are the supporting cells. So let's take a look at them. There's 4 major types in the central nervous system. I think I can erase this. Just kidding. We'll call it neural tissue, and we'll say CNS, ENS, and we'll say we have nuclei. I'm just going to add the words in here, and you can go in and fill them in later. Right, nuclei, we have tract, and we have neuroglia. And here we had nerve and ganglia and neuroglia. So, the types of neuroglia in our central nervous system include something called an astrocyte. An astrocyte is a small star-shaped cell, right? Think astro, like astronomy, right? It's literally a star-shaped cell. Oligodendrocytes. Microglia. And ependymal cells. And in our periphery, we have Schwann cells and satellite cells. So, astrocytes are there for anchoring. Astrocytes are these small star-shaped cells that help form something called the blood-brain barrier. They isolate and protect the brain from blood. And I'm just going to abbreviate BBB up here for blood-brain barrier. We'll talk more about what the blood-brain barrier is when we get into protective things. But that's what astrocytes do. They kind of lock in, those little star shapes kind of interlock, and it forms this big barrier around the brain so that blood doesn't mix with it. If you were to, if I were to like do brain surgery right here in front of you and chop someone's head open, or anything, or an animal, right? Chop their brain open. Once we got past the bone and the coverings, there's no blood in there. There's no blood in your brain. There's juices. It's the cerebral spinal fluid, but there's no blood in it, right? Because those astrocytes create this blood-brain barrier that separates the blood from the brain. Oligodendrocytes wrap around the axons of our neurons. So they're kind of like these insulating protective blankets, but they're cells that wrap around the axons. When they wrap around the axons, they form something called myelin. Myelin is there to help speed up action potentials. It helps the communication go fast. It's like pushing the turbo button. Yep, most action potentials are lightning fast, but when you put myelin on them, they're like the speed of light. Not really, but they're very, very, very fast. Okay, myelin is the turbo button on that action potential speed. When our axons have myelin, they're going to send that action potential much, much quicker. And we'll look at how that happens eventually. Microglial cells, these are actually immune system cells. They're phagocytic. They're there to gobble up any debris or invaders that don't belong. So they're part of the immune system. We call them phagocytes. Right? A phagocyte is an eating cell, a cell that eats, like Pac-Man. Ependymal cells are cells that secrete cerebral spinal fluid. They have cilia to circulate the CSF. So cerebral spinal fluid is the juice that surrounds the brain. And these ependymal cells have blood on one side, right? And then they have the cerebral spinal fluid on the other. They're pulling the nutrients out of the blood and putting it into the CSF and doing the reverse, pulling the waste products out of the CSF and putting it into the blood. Meanwhile, they have cilia kind of beating around and waving around to move that CSF and circulate the CSF through the central nervous system. So there they all are. In the periphery, our Schwann cells act like the oligodendrocytes. So these provide the myelin in our periphery for that quick action potential. And satellite cells help provide support for the cell body. So that's kind of what it would look like. Satellite cells kind of guarding the cell body, Schwann cells surrounding the axon. So let's talk about that myelin. So this process of myelination, or growing myelin, becoming myelinated, is going to help speed up action potentials. And it's kind of like, again, going back to the electrical wiring, if you have certain types of wires in your house, they're going to send signals faster or slower depending on what type of metal they're made out of. So like aluminum wires, and I know this because we rewired a house before, aluminum wires conduct really slow, they are slower, right? So they're not as quick. That would be like an unmyelinated axon. It's going to send a signal, it'll get the job done, but it's going to be slow. A copper wire, though, can really get it done, right? Copper wires are the faster way to conduct the electricity. Myelinated axons are the faster way to conduct an electrical impulse. And it's going to do this by insulating that axon. That insulation allows for the action potentials, this conduction speed, to be much, much quicker. Other things that are around the axon, there's something called a neurolemma. The neurolemma is the just the outer surface of our axon. The number of axons that are myelinated can vary between central nervous system and peripheral nervous system. And when myelination happens can really affect how our neurons work as well. Go ahead.
What's the difference between the neurolemma and the Akzo Lemma?
Okay, so the neurolemma is in the peripheral nervous system. So this is, yeah, so it's literally going to include, so if this is a peripheral nervous system, and we have a Schwann cell here, and a Schwann cell here, and a Schwann cell here, the neurolemma would be another layer on top of it. Yeah. Yeah, and you'll see it in lab on the model. There's like a piece of like connective tissue like surrounds it. Right. So this is showing you that myelination process. When myelination happens, will affect development. When babies are born, most of their axons aren't myelinated. And if you've ever watched a baby, like a newborn, move, their movements are very like jerky. They're kind of like kick and, you know. And they also don't have the coordination, right? Have you ever watched them like try to find their thumb or something to suck on? Like they'll just kind of be like they can't find their thumb. It's not because they don't know where it is. It's because they physically don't have that fast, rapid communication because their axons aren't myelinated, right? They start to myelinate, they can find their thumb. They start to myelinate, they can slowly learn to hold themselves up, right? When toddlers start walking, they fall down all the time, right? Because they're not myelinated. As they use those axons need to become more and more efficient at sending those impulses. Myelination is laid down and they become better at walking, skipping, walking upstairs, all those things, right? So, kind of cool. All right, that's just showing you all the myelination stuff. So, myelination produces things called nodes and internodes. Nodes are the unmyelinated spaces. So the action potential is going to be jumping from node to node. Instead of having to travel down the entire axon, it only has to jump from node to node. The pieces where there's myelination, where there's a Schwann cell or oligodendrocyte, is called an internode. This is where those neuroglial cells are internodes, and nodes are the unmyelinated axons. That's where that action potential is going to be jumping. These myelinated fibers is what creates white matter. Have you heard of that before? Gray matter and white matter when talking about the brain. So gray matter is unmyelinated axons and cell bodies. White matter is myelinated axons. So when we look at I'll just draw it here. When we look at our spinal cord, a cross section of our spinal cord, this is, a transverse section looking into the spinal cord. Your spinal cord actually looks like this. Well, it doesn't because this is a horrible drawing, but there's gray matter on the inside and white matter surrounding it. This white matter are the tracks, right? Tracks of axons coming up into the central nervous system, going up into the brain or going out to the periphery. All of this is where all the cell bodies are, right? White matter is the myelinated axons. On the brain, guess what? It's reversed. if I were to draw a cross-section of the brain, I'll try. Draw a cross-section of the brain. This is your brain on drugs. I'm really bad at it. Okay. This is a sagittal section of the brain. The way the brain looks is it has gray matter superficially and white matter on the inside, right? All the cell bodies are at the top and are around because they're all projecting down into the brain, to other parts of the brain, to the spinal cord, right? Because the white matter are the actual myelinated nerves, right? They're the myelinated tracks going in and out, the highways, right? Carrying information in and out and around. So they're put together kind of reverse, right? Part of that's protective, right? Our brain is much more protected than our vertebral column is and our spinal cord, right? But our brain also has all of these cell bodies surrounding because all of those fibers are projecting in, right? That's the white matter. And they appear white. The myelination makes them appear white. Some of our cells are able to regenerate. The central nervous system cells cannot regenerate. Only certain cells in the periphery can. And even there's like an asterisk. Like it has to be surrounded by enough satellite cells and Schwann cells to actually regenerate. So if you've ever known someone who's been in a really bad car accident and have nerve damage in their arms or legs, right? Not talking spinal cord nerve damage because that's a different ballpark, right? That's usually irreversible. But if we're talking in the periphery, those nerves can slowly regrow. They can slowly regrow. Maybe you had a really bad car accident, your arm was crushed in the car accident, you had to have part of it, grafted back on or whatever, right? All of those, you lost feeling. Eventually, the nerves can regrow, only if there's a certain amount of neuroglial cells there. So regeneration takes place in a few steps. It's not just like, hey, we're just going to regrow a nerve here, right? What actually starts is the axon itself, and I'll use this as my example. The axon actually, let's say that it was broken right here. All right, we have a damaged axon. The distal axon disintegrates. It's called Wallerian degeneration. It just goes away, right? And so what we have left is basically a stump of an axon. Well, I'll erase that black part too. The neuroglial cells kind of extend, and they're basically like mowing a path for the new axon. These neuroglial cells extend, and then the axon can kind of grow. And then they extend some more, and then the axon can grow. And then they extend some more, and then the axon can grow. Right? So this is the process of regeneration, but it only takes place in the peripheral nervous system. It's very slow. It's called the generation tube, is what it is. This regeneration tube that gets laid down. And it's only going to happen if there's a certain amount of those neuroglial cells there. The new axon will eventually connect with its target cell where it needs to be. This is a picture of it if you couldn't follow my little animated drawing up here, right? So we have some type of injury, Wallerian degeneration, breaking away. Doesn't want to zoom in when I want it to zoom in. Anyway, it's going to wear that away. Our Schwann cell elongates, forms that regeneration tube, and the axon can grow within it. Nice and protected. So, there are cancers of these tissues, right? Brain tumors, gliomas, and astrocytomas. So, primary brain tumors usually originate from these glial cells, from the neuroglial cells. They're called gliomas. They have a much higher rate of division. Your central nervous cells don't divide. The brain cells in